A key element of the UK Spine's mission is to support development of drugs which target biological drivers of aging. Toward this goal, the UK Spine's Bridge program has funded the project Identification of Novel Epigenetic Targets to Reactivate Exhausted and Senescent T Cells. The principal investigator is Dr. Annette von Delft of the Centre for Medicine's Discovery at the University of Oxford. Collaborators on this work are Professor Eleanor Barnes of the Translational Gastroenterology Unit at the University of Oxford and Professor Janet Lord, Director of the Institute of Inflammation and Aging at the University of Birmingham. Additional contributors include Professor Paul Brennan of the University of Oxford and Dr. Charlotte Green of the Drug Discovery Unit at the University of Dundee. T cells, which play a key role in the immune system, suffer age-associated functional decline. Populations of senescent and exhausted T cells, characterized by decreased effector function and proliferative capacity, increase with age. The diagram is from a 2014 Frontiers in Immunology review article and depicts observable characteristics of senescence and exhaustion in T cells. This decline in T cell function compromises immunity. Toward reversing this decline, T cell reactivation is being explored. Prior efforts focus on identifying targets for checkpoint inhibitors, but not all patients respond, and relapse is common in those who do. Checkpoint therapy has limited impact on T cells' epigenetic signatures, indicating an opportunity to establish a more robust response via complementary therapy which acts on epigenetic targets. The research team has access via the Structural Genomics Consortium to a library of 54 potent and selective epigenetic inhibitors. These will be screened for ability to reactivate T cells. For lead hits, the mechanism of action will be interrogated and translation initiated, supported by early engagement with translational partners. The initial stage involves development of screening assays for exhausted and senescent T cells via FACS panels designed for this work. Next, pooled human PBMCs are treated with the epigenetic inhibitors. Cell donors are age 65 plus with both healthy and chronically infected cohorts. Lead hits are then identified from extra and intracellular panel data. These hits will be interrogated for mechanism of action, with ChIP-seq and ChIP-PCR data providing insights on alteration in epigenetic regulation and killing assay data reflecting activity level. With early input from downstream collaborators, lead hits will be explored as potential candidates for further drug discovery efforts. To this end, the Drug Discovery Unit at the University of Dundee has been engaged for early-stage discussions. Separate from this specific drug discovery translational pathway, the broader research community can leverage the project data in their own drug discovery efforts. In addition to traditional knowledge exchange outputs, this project has yielded a new collaboration between different labs at the University of Oxford, and also between the University of Oxford and University of Birmingham. Early engagement has been established with the Drug Discovery Unit at the University of Dundee, both to optimize project design and to initiate a dialogue on potential translational pathway. The project also shares data and methods with the broader research community, consistent with adherence to an open science approach.